So let's go ahead with Alina, I'm sorry. Alina, you're here, honey?
Yeah, I'm here.
Okay, let's go with your presentation. Let me just yeah, go ahead. Start sharing.
Can you see my screen?
Yes, sweetheart, yeah.
Okay. So, hi, my name is Alina Shinde, and I'm a sophomore at the Edison High School STEM Academy in New Jersey. And my presentation is on the genetic modulators of prion-like protein propagation in Alzheimer's. So, like I said, my area of investigation was investigating the genetic modulators, especially through hereditary or familial Alzheimer's disease, and their effect in tandem with prion-like protein propagation in the form of tau and beta amyloid in Alzheimer's disease. So, my questions that guided my research were first, what genetic variations contribute to the prion-like behavior of tau and beta amyloid proteins in Alzheimer's? And then, how do familial Alzheimer's disease or FAD genetics influence the spread of tau and A beta pathology? And can targeted interventions on the cellular prion protein FAD genetics, tau and A beta, used collectively disrupt the progression of Alzheimer's more effectively than another method? So this research is definitely significant because understanding the genetics of AD is crucial for developing personalized treatments and interventions to prevent it or mitigate its effects. And the hereditary background of AD often goes overlooked because FAD is more rare than sporadic AD, where FAD is hereditary and passed down through families, and sporadic AD is found later in life and just comes up in people randomly. However, these hereditary genes are often associated with and seen in epigenetic alterations in sporadic AD and can be used as a more accurate starting point for future targeted therapies in both types of Alzheimer's. So, because my paper was a literature review, I'm just going to be going over the background and current accepted theories. So, first, the first section of my paper involves the prion like properties of tau and beta amyloid. So, tau and beta amyloid are proteins that exhibit prion like behavior, which is the ability of certain proteins to adopt a misfolded conformation that can induce the misfolding of their normal versions. And they propagate and aggregate into neurons and different cells, which results in dysfunction, especially in Alzheimer's disease. And this is pivotal for targeted therapeutic interventions. So, the first accepted theory in the scientific community related to tau and A beta is tau propagation, which basically says that misfolded tau spreads between neurons, initiating neurodegenerative cascades. And the second accepted theory has to do with the beta amyloid cascade, which says that there are sequential events triggered by beta amyloid aggregation causing synaptic dysfunction and neuronal degeneration. And the second part of my research had to do with how FAD genetics is implicated in protein misfolding. So, FAD is essentially Alzheimer's disease associated with specific genetic mutations. And research on this type of Alzheimer's shows that mutations in key genes link genetic factors to the misfolding of crucial proteins like tau and A beta. And this underscores the need to explore genetic interventions for Alzheimer's. So for my methodology, like I said, I split it into two sections. First, starting with the genetic analysis of FAD and analyzing key genes with specific cases, and then seeing how that is related to protein misfolding in terms of tau and A beta. So I explored molecular mechanisms connecting genetic factors to this protein misfolding and showed that the interplay of familial genetic factors and protein misfolding lead to this cascade of events where FAD associated mutations actually amplify the prion like behavior of tau and beta amyloid. So, more specifically, I discovered mutations in MAPT or the tau gene, presenilin genes 1 and 2, and the APOE genes in FAD affected families. and then correlated these mutations with increased susceptibility to prion like processes. And overall, these play a crucial role in the aggregation of beta amyloid and the abnormal phosphorylation of tau. And finally, at the end, I established the possibility of stem cell therapy and immunotherapy, as well as combination therapies that target these genes and proteins. And these involve the expression of anti tau and anti A beta antibodies or peptides. And then another form of therapy would be to inhibit activators that promote tau phosphorylation or A beta production. And more specifically, these have to do with kinases, which um, like start the phosphorylation in reactions and cellular processes. So I would target GSK or CDKs. So, like I said, for my results, FAD associated mutations amplify the prion like protein properties of these proteins. And overall, this contributes to research on Alzheimer's because it lays the foundation for developing targeted therapies that address both the genetic and protein related aspects, which are often overlooked as hereditary Alzheimer's is much rarer than sporadic AD. And the limitations for this research was that we also have to take into consideration other factors of Alzheimer's, such as environmental and epigenetic factors. And this also has to do with individual variabilities, which can, um, you would want to use like personalized therapies rather than just one set for the general public. And then finally, we also have to, of course, take into account ethical considerations, especially because I propose stem cell therapy, um, which has to do with genetic modifications, which always have ethical controversy attached to them. So, for my research experience, I wanted to thank Dr. Varkey and Professor Virgil, who both helped in creating this paper and um, narrowing down the content into what was important. So, specifically, Dr. Varkey guided my exploration of genetic factors in Alzheimer's and helped with understanding these complex molecular pathways. And from the very beginning, provided insights into prion like behavior, which shaped the direction of my research from the beginning of the program. And Professor Virgil offered feedback which helped refine my paper's content and assisted in synthesizing my research findings into a cohesive paper. And for the overall research program, I had a great experience because I learned the methods of research and how exactly to write a research paper, and also gained a good understanding of advanced analysis techniques and their application in neurodegenerative research. So, yeah, thank you for listening to my presentation.
Amazing. You want to just stop sharing so
Yeah.
that Dr. Werkey can ask you a question?
Yeah.
Now, Alena, again, like very comprehensive study, a lot of data. I can see that you went into detail of the different genes involved in Alzheimer's and in general, like neurodegeneration. So, also like what you have looked into is something way into future, which is what people are trying to get to, that is personalized medicine. And especially in case of Alzheimer's and these neurodegenerative diseases, it's very apt because sometimes the epitope or the antigen which is present, it's probably different in different individuals. So you need different therapies or different antibodies which might work for one person, which may not work for another individual. So
Yeah.
really good job. So I have a question. So let's say with every antibody or every therapy, there is like some kind of side effect associated with it, isn't it? Uh,
Right. Yeah.
okay.
So,
So let's say if we have
Hmm.
two different therapies, do you think that the side effects will also be amplified in that case?
like, you mean depending on the therapy, could side effects
Yeah.
be different? Well, because these therapies are so like not developed yet, my research was very brief and only went over like what could possibly be done. As for
No,
side of
I just want you to guess. That's all.
yeah, I would say that there probably would be differences, especially because
Uh-huh.
these are catered to different people with different health, um, different diet, different lifestyles. You
Yeah.
definitely wouldn't have the same side effects in each person. So that would, of course, have to be studied further.
Good answer. Yeah. So apart from like when you give like a personal medicine, the side effects also could be different for different individuals
Yeah.
depending upon their genetic background. Okay. Good job.
Thank you.
Yeah, great job. A round of applause for Elena.